Hello students, welcome to the EPG Pathshala. My name is Dr. Rajeshwari Sinha and in today's lecture we are going to learn about Ramachandran plots. Let us see what objectives are we going to address in this lecture. At the very outset, we are going to understand the different torsional angles. We will look at what is Ramachandran plot and also know about the different torsion angles around peptide bond in proteins. Further, we will understand the different regions on the Ramachandran plot, learn about the allowed and disallowed conformation of proteins and also the applications of Ramachandran plots. We hope you have an enjoyable time listening to this lecture. Introduction. The covalent backbone of proteins contain thousands of individual bonds around many of which free rotation is possible. Proteins can assume myriads of conformation. The term conformation is defined as the spatial arrangement of atoms in proteins. Any structural state that can be achieved without the breakage of covalent bonds constitutes the most possible conformations of a protein. Several parameters are involved in completely describing a polypeptide helix or a beta pleated sheet. The backbone of protein is a linked sequence of right planar peptide group. The backbone conformation of polypeptide is specified by the rotation angles or torsion angles about the C alpha nitrogen bond, which is phi, and the C alpha carbon bond, which is psi, each of which its amino acid residues. The plane of the peptide bond can rotate 180 degrees in either direction, plus 180 degrees to minus 180 degrees, then one can begin to construct a graphical representation of all the psi and the phi rotations about the peptide bond. In this slide, we have a look at the schematic representation of the various types of psi angles. Psi can take values from 0 to 180 to 90. In the next slide, we look at the various possible schematic representation of the phi angle. Phi can also take values from 0 to 90 to 180. Importance of torsion angles. Torsion angles are among the most important locale structure parameters that control protein folding. These angles provide the flexibility required for the polypeptide backbone to adopt a certain fold. The third possible torsion angle within the protein backbone is called omega and is essentially flat and fixed at 180 degrees. This is due to the partial double bond character of the peptide bond, which restricts rotation around the CN bond, placing two successive alpha carbon and carbon, oxygen, nitrogen and hydrogen between them in one plane. Ramachandran plot. Rotation of the protein can be described as rotation of the peptide bond plane relative to each other. Allowed values of phi and psi are graphically revealed when psi is plotted versus phi in a Ramachandran plot. Ramachandran plot was originally developed in 1963 by G. N. Ramachandran, C. Ramakrishnan and V. Sasi Sekharan. G. N. Ramachandran and his co-workers used computer models of small polypeptides to systematically vary phi and psi with the objective of finding a stable conformation. In this slide, we have a look at one example of a Ramachandran plot. We see how the psi angle and phi angles have been plotted against each other, where each of them move from minus 80 to 180. And we also look at the stable conformations achieved at different angles of the beta sheet and the alpha helix. Regions of Ramachandran plots. The Ramachandran plot helps with determination of secondary structure of proteins. Quadrant 1 shows a region where some conformations are allowed. This is the region where rare left-handed alpha helices lie. Quadrant 2 shows the biggest region in the graph. This region has the most favorable conformation of atoms. It shows the sterically allowed conformation for beta strands. Quadrant 3 shows the next biggest region in the graph. This is where right-handed alpha helices lie. quadrant 4 has almost no outline region. This conformation, psi around -80 to 0 degrees, phi around 0 to 180 degrees, is disfavored due to steric clash. This slide is a complicated version of the Ramachandran plot. It shows where different kinds of secondary structure of proteins lie in the Ramachandran plot. We can see where the anti-parallel beta sheet lies, the parallel beta sheet lies, the collagen triple helix, the left-handed alpha helix, the right-handed alpha helix and the closed ring, all of which find different positions within the plot based on the torsion angles. The phi and psi angles of different secondary structures are shown in the table below. For example, we see that the right-handed alpha helix has a phi angle of -57 degrees and a psi angle of -47 degrees. Likewise, different types of secondary structures have different phi and psi angles. In this slide, we see what different nomenclatures are adopted for the Ramachandran plots. So students, we hope you are finding this lecture useful. We have now reached midway into this lecture. We understood the concept of phi and psi angles and the importance of torsion angles. Next, we understood what are Ramachandran plots, what do the 4 quadrants of the plots mean, and also saw some representative examples. Now, it is time for us to see where and how these Ramachandran plots are applicable and useful. Let us take a look at these. Ramachandran plot and quality of protein structure. In instances where the protein structures are obtained from X-ray crystallography are not properly refined and principally for wrong homology models, the torsion angles presented in disallowed region are determined from the Ramachandran plot. It is extensively used in evaluating the quality of experimental protein structures or structures built through homology modeling. A number of tools are available for constructing Ramachandran diagram of a query protein. Such tools include Rampage, ProCheck, Moleman2 and MolProbity. The applications of constructing Ramachandran diagrams are as follows. For validation of crystal structures, validation of macromolecules crystal structures starts from the 1990s. It was an idea in the beginning. The plot was used to check the targeted function in the crystallographic refinements. The validation studies can be easily performed with the tools like ProCheck. For reproducibility, quality measures has been taken as a core, allowed and generously allowed regions. They are commonly known to have reduced occurrence of errors. Ramachandran plot found very helpful and useful in the model building and methodologies building enhancement. Validation of crystal structures using ProCheck. Various bioinformatic tools are available online to check protein structures for its stereochemical quality and accuracy. One of the most such versatile tool is ProCheck, which verifies the structure by generating a number of postscript plots describing its overall as well as residue by residue geometry by comparing the stereochemical parameters derived from well-refined high-resolution structures. This tool is available online free of cost. Basically, it checks the deviation of geometry of the residues in a given protein structure from normal or unusual. The regions that are highlighted as unusual regions by ProCheck are not necessarily errors as such, but it means that these regions are required to be checked carefully. The input to ProCheck is a single PDF file containing the coordinates of the protein structure of interest. This program firstly cleans up the coordinate files with the motive of correcting any mislabeled atoms and creates a new coordinate file which has an extension of .new. This file contains the atoms labeled in accordance with the IUPAC naming conventions. The output comprises of a number of plots such as Ramachandran plots, Gly and Pro plots, Chi1, Chi2 plots and distorted geometry plots. The plots can be in color or black and white. Applications of Ramachandran plot. A number of tools are available for constructing Ramachandran diagram of a query protein. Such tools include Rampage, ProCheck, Moleman 2 and Molprobity. Other applications of Ramachandran plot include validation of crystal structures, structure prediction and design, and lastly, selecting and filtering the top data sets. Students, we have now come to the end of this lecture. Thank you for being there with us. Let us summarize what we have learnt in this lecture. The relationship between the amino acid sequence of a protein and its three-dimensional structure is a convoluted puzzle. An understanding of the structure is essential to the study of the function of proteins. The nature of the covalent bonds leads to constraints on the structure of polypeptide backbone. The C-alpha nitrogen bond and the C-alpha carbon bond can rotate to derive different bond angles. The allowed values for these angles can be determined through Ramachandran plot. Thank you for watching this lecture on Ramachandran plots.